site of the crime, it can be used to detect the DNA of the criminal and compared with that of the suspect to ascertain the truth. So, do you know there is an Indian scientist named Dr. Hargobind Khurana who works in the field of the molecular biology, and he got Nobel Prize for his work. So, Hargobind Khurana was the creator of man-made gene. It is a matter of pride that Nobel laureate Dr. Hargobind Khurana, who was born in our country, synthesized an artificial gene in the lab for the first time. For this, he got Nobel Prize in 1970 for his contribution to molecular biology. So, now we will talk about the DNA molecule. DNA molecule is a polynucleotide. It is made of units called nucleotides, each of which contain one nitrogenous base, a deoxyribose sugar and phosphate. So, there are 4 nitrogenous bases: adenine, guanine, thymine and cytosine, and hence we can say 4 kinds of nucleotides in a DNA molecule, and various combination of these nucleotides in a segment of DNA form the different genes. In physical structure, a DNA molecule is a double helix containing 2 polynucleotide strands. So, now we talk about the DNA replication. The cell division takes place in a manner so that one cell divides into 2 identical cells at the same number of identical chromosomes. So, therefore, prior to cell division, every chromosome should contain 2 chromatids made of 2 identical DNA molecules. This is achieved through the process of DNA duplication or DNA replication. So, major step of DNA duplication or replication are: prior to the cell division, we know every chromosome should contain 2 chromatids made of 2 identical DNA molecules. So, one double stranded DNA molecule unwinds with the help of certain enzymes to expose 2 strands of DNA. DNA polymerase enzyme catalyzes the formation of a new daughter strand, which can form a double helix with 1 strand of parental DNA molecule. So, 2 DNA molecule, each with a parental strand and a new strand gets generated. So, 2 identical DNA molecules then become 2 chromatids which remain attached by a centromere. Thus, upon DNA replication, each chromosome contains 2 identical molecules of DNA housed in 2 chromatids, and during cell division these 2 chromatids separated out as 2 chromosomes, one each